Hello, everyone. Thank you so much for coming to our symposium. Thank you for the speakers for your interest and participation in speaking at the symposium. And thank you for the meeting organizers for helping us put together this wonderful 2 sessions on correlated adaptation. My name is Shirley. I'm co-organizing this symposium with my advisor, Dr. Anurag Agrawal. And ever since the meeting organizers put up this call for proposals to do a symposium on whatever we want, we jumped at this opportunity to do a really focused but very cross-disciplinary symposium on correlated adaptation. And as you can see, I'm one of the co-organizers. Anurag, my advisor, unfortunately cannot be here, which means that I can do whatever I want. And I have to take this opportunity to put up an embarrassing photo of him. So he cannot be here because of a very unfortunate last-minute scheduling conflict. This is a photo of him grimacing a little bit in my greenhouse. He was overcoming extreme physical pain in that photo to try to look at a really fun, unexpected floral mutant that I've grown up in a greenhouse for the project that I'm going to present in this afternoon. And the point of this photo, it's not really that embarrassing. The point of this photo is to really try to convince you that he would overcome extreme physical discomfort to show up for you and support and look for good science. And the fact that he's not here means that he really cannot be here. And the second thing that I get to do in his absence is I get to talk about his work. I think those of you who are professors or who have trained students have the distinct pleasure of being able to talk about your trainees' work, but rarely do you get to do the opposite. And so for the past 15 years or so, the focus of the lab has been the co-evolutionary ecology between the toxic milkweed plants and the community of the specialist herbivores that feed on the milkweed plants. And so a few weeks ago, our lab has synthesized about 10 years of conceptual thinking and empirical comparative work and published in MNAT on this synthesis of what he calls the sequestration defensive syndrome. And so in the milkweed plant, the plant synthesized a cocktail of chemical defenses in association with a lot of physical defenses to protect itself from being fed on by herbivores. And the community of specialist herbivores in return have co-evolved these strategies going from the enzyme level to the behavioral level so that they can feed on and really feast on these toxic plant and turn these toxins inside their body as a defense against their own predators. And so these kind of associated traits in these insects that have evolved together, he's been thinking about these traits integratively as kind of a collective package of defense strategies that he calls syndrome. And this paper explores how the steps and sequences of, the steps by which the enzyme adaptation, the behavior adaptation, and the other kind of adaptation evolve together, how repeatable it is across all arthropods, what are the exceptions, and what are some of the generalities. This is a really good paper that I highly recommend you check out. As a student in his lab, I do not study plant and herbivore interactions, but I'm broadly interested in floral evolution. So this is where this shameless plug comes in. This afternoon, I will be presenting in a regular session on how different floral traits evolve. And by different floral traits, I mean the different modalities in which flowers can advertise itself through visuals, rewards, and volatiles, and how these different traits can function together for the purpose of attracting pollinators. And while the specific questions that the two of us study might differ, there is something that unites our conceptual interest, which is we're both interested in how adaptation in a whole organism, despite them being discrete traits, function together to the same selective environment. And we've been colloquially using the word syndrome in each of our work to refer to this set of correlated adaptation. And as you can see, we've published with those words, but it's come to my realization that the word syndrome has been really provocative in that it evokes very divisive feelings among reviewers and audiences who hear these words. It's kind of like describing a species as basal to systematists or phylogeneticists. People who understand what you mean by those words kind of just accept it and move onward. Other people who really advocate for the clarity and precision of this word really is ready to engage in a debate. And so, as you can figure, Us co-organizers are a huge proponent of using the word syndrome, or really the conceptual reasoning behind using the word syndrome, which is we really need to think about trait adaptation in an integrative way, in that all trait, not a single trait usually doesn't suffice for the purpose of adaptation in a novel environment. So multiple traits evolve together for the same selective environment, and therefore we have to consider the evolution of all traits together in order to understand the evolution of a single trait. And so for this brief intro, I'd like to go into a little bit into why is it worth it? Why is it worth it to organize this dedicated symposium just to think about how different adaptation may evolve together in a correlated fashion? And I think to really answer the questions of why to study correlated adaptation, we really need to think about why to study adaptation at all. And I think regardless of the discipline and the system you come from, I may say for all of us that we are united by these overarching questions of trying to understand and organize the diversity of life. Many of you, like me, might come from a system that is more multicellular and eukaryotic, and that is really represented by that small green arrow pointing down there. And the diversity we have in there is already staggering. So as a main mission of biologists, ever since the birth of the field of evolutionary biology, really is how do we organize it? And a quote, and I think summarizes the spirit of that mission really well, is this MacArthur quote that says, "To do science is to search for repeated patterns, not to simply to accumulate fact." While I think many of us who are naturalists do take refuge and find it really fulfilling to describe the diversity of life, because there's so much of it out there, it's a very rewarding mission of a scientist to look for patterns that may explain and organize so that we might anticipate and explain the new diversity that we encounter out there. And I think historically, there's been two organizing principles in trying to explain this diversity of life. One is that we know that shared origins may generate similar lineages. So organisms, we might organize and explain the diversity because they're more closely related. Where I'm from, in New York State, in about a month or so, if you take a casual walk across any old field, you will see a consortium of different goldenrod plants in the field. The photos that I'm showing up here are 8 photos of 8 different species. Even if you're not a trained botanist, you will at a glance recognize that these are very closely related species, and there's something uniting them more so than any of them is to other Asteraceae in the field. So that instinct of picking apart the similarities and differences is kind of driven by the result of this historical evolution that made lineages more similar to each other if you're more closely related. But the traits that unite goldenrods and the traits that unite Asteraceae have only evolved once, right? Because these evolutionary events, the emergence of Asteraceae has only happened once, and the rules, the patterns and processes in these do not explain what makes maples a maple and what makes oaks an oak. And so the second organizing principle actually lies in the quote that I just showed you, which is the word repeated. We don't want to just look at things that happen as an evolutionary singularity. We want to look for patterns that happen repeatedly across the tree of life. And that's when we really come to take advantage of convergences, these gifts of comparative biologists that allow us to look for similarities in distantly related lineages that find themselves in the same selective environment. So if we move south of Montreal and beyond New England into the desert, you will find a community of succulent plants that have evolved varying adaptations to arid and hot and dry environment. And unlike the goldenrod photos that I'm showing you previously, these 8 plants all come from different plant families, but they've converged on the same habitats and growth form so that they can adapt to the shared collective environment. And their similarity does not rest just on superficially looking similar. They've converged also on their fundamental strategies to photosynthesize, which is what essentially makes a plant plant. In response to arid, low CO2, atmospheric CO2 and hot climate about 30 million years ago, many different plants evolved this CAM or C4 photosynthesis, which is a way to adapt to this really harsh environment where photosynthesis get repressed because photorespiration comes in. And so you can see that these convergences happen at multiple levels, from the way that a plant looks to the biochemical and the cellular and genetic mechanism of dealing with the novel environment. And all of these have to happen at once and integratively in order for these lineages to survive and diversify in these novel environments. And so really, recently, we've been advocating more for integrated consideration of evolutionary change. Still sticking to the example of adaptation to arid and hot environment, the adaptation can happen on the overall form where there's broad life history and growth form adaptations such as perenniality, huge root systems, the really compressed internodes that allow you to conserve water, water loss. The adaptation can happen at the within organism trait level, such as the evolution of wax, the evolution of highly reduced foliage. And it can also happen at the biochemistry and the cellular level, where you reorganize your leaf cell anatomy so that you can allow for these photosynthesis and C4 photosynthesis to occur. And of course, for those of you who are gene people, genomic adaptation, a whole cluster of genes have to evolve for these biochemical processes to happen. And trade-offs, constraints, and synergy permeates all level of these adaptations, right? Because these different adaptations at different levels have to function together, they have to evolve together, and sometimes not in a straightforwardly correlated fashion. Sometimes not all traits can evolve together because of genetic or ecological constraints. Sometimes they synergize, and certain discrete trait value have to happen together in order for any trait value to work. And this kind of exceptions and generalities to how traits evolve together or not has been a really fascinating focus of our research, and I hope many of your research for the past years. And hence this symposium. And so in getting together a symposium from many systems and many questions that look at trade-offs and synergies, we're hoping that we inspire a lot of thinking and discussions and really appreciate how the study of adaptation has, even if you're only studying one trait, has to take into consideration of all traits. And we've been very intentional about trying to gather speakers from all systems because we've really benefited from cross-pollination of studies from different systems. So several decades ago, the book of Island Geography has first proposed the idea of R and K life history strategies in animals. While that has been an incredibly successful way of thinking about life history strategies in animals, several decades later, plant biologists have co-opted the thinking and the conceptual generalization from that book, and then come at our own understanding of how we see parallels of that kind of change in plants on islands. And we've had actually quite a few talks so far in the meeting on correlated adaptation and convergent evolution in other sessions. And I've been really awed by the kind of new questions that we've been able to ask and answer with new technology and new methods. So I'm putting up a few questions here that I'm hoping that you guys may take in mind as we go through two sessions of really amazing talks across systems and across questions. Some of it might be that while the eventual phenotype might show patterns of correlation and adaptation, how do we get there? Are there trade-offs and synergies on the steps of evolution leading up to those phenotypes that might add to the richness of our understanding of the diversity? Can trade-offs and constraints be overcome? Are these trade-offs and constraints, if you give it more time and more genetic diversity, can we overcome what we thought of as unbreakable trade-offs between traits and genes? And because this is an introduction talk, I'd happy to discuss some of the conceptual thinking behind after, and the first talk will shortly begin.
Howdy, I'm Jonathan. Just a quick note, slight disagreement between the online and in-person schedule, so if you're here for Dave's talk, it will be in 15 minutes. I'm a graduate student in Mark Rauscher's lab at Duke University, and I'm really excited to be talking to you guys today about how flower color may modulate selection on other floral traits in the Epimea cordata triloba. So, like many of you all, I love the diversity we have of all organisms around us, right? And in particular, we see among a lot of these organisms interesting traits, right? And interesting ways that traits have kind of come together to make these cool features that we appreciate, right? Now, there's been a lot of ink that's been poured over how these traits can be correlated, right? And a lot of it has to do with constraint. And there's been a lot of great examples about how constraint can frame correlated selection. For example, we have this graph over here that, time to first flower and size at first flowering. You can't have both, right? You've got to necessarily sort of have a trade-off between one and the other. But I've also started to get curious about if selection can, selection on certain binary traits can generate correlations. And so I'm going to kind of walk you through what that might look like. So unless you have super keen eyesight, you might not see that one of these faces here is a smiley face and one is a frowny face. And, you know, you might, if I tell you you're going to get something really good if you choose a smiley face, something bad if you choose a frowny face, you might sort of be choosing at random here, right? But if we change a different trait, like the color, we can now see which one is a happy face and which one's a smiley face. Now note that color itself isn't going to give you good things. It's just the ability to perceive color is letting you choose the big smiley face. And I think this is hopefully an apt analogy for thinking about how a change in a certain state trait, like color, can actually change patterns of selection, right? You can choose the happy face when you have this pink color, but not when you don't. When we're trying to think about these two different possibilities of what can be driving correlated selection, I think flowers and floral systems, not just because they're so beautiful, are a great, they're a great system to sort of try to explore this, right? So not only do we have tons of covariance among a number of different flower traits that have, there's been great literature about all the different constraint that has sort of modified these pollinator syndromes. We also have just beautiful flower colors, right? And we oftentimes have systems where difference in flower colors change how pollinators visit plants. And so I think we have a great system in this in Morning Glory. There's been so much amazing work done in Morning Glory. So I'm going to be talking about these two species that have recently diverged. One is a mixed mater, and one is a highly selfing species that has experienced selection towards selfing. It has white flowers, smaller flower size, less nectar, less concentrated sugar. And because there's been so much great work that's been done in the system, we have a lot of really great tools to use, including recombinant inbred lines. For those that aren't familiar, recombinant inbred line, you take both species, in this case, Chordata triloba and lacanosa, you cross it to make an F1, you self-bet for a number of generations, and then you have your lines that have these mixed genetic backgrounds. So in the system, we have lines that we've scored for flower color, and on top of that, previous work in the lab has measured a bunch of different flower size, sugar concentration, and seed traits. And when previous researchers did this, they showed kind of an interesting result, that within these nectar traits, we see high levels of genetic correlation, and within size traits, we see high levels of genetic correlation, but between both categories, we actually see pretty weak genetic correlation and trait correlations. The genetic, this is a trait correlation graph that you see, but the genetic correlations are very similar. So the one piece of data we just don't have yet is fitness estimates, right? So if we want to understand how, what's going on with size and sugar concentration in terms of success in correlated selection, we need to have some estimates of fitness. Now, interestingly, because we know size and sugar are weakly correlated, I would not expect constraint to play a large role in selection for these traits, right? So this is where I come in. I'm an evolutionary ecologist, and I work with these lines in the field. This is my field site out in Whiteville, North Carolina. And I planted, in 2021, I planted 16 replicate plots that had 50 arrays, 25 of each color, and 25 lacanosa as a control. During the course of the season, I let them grow. At the end, I bagged the plants and took them in a U-Haul back over to campus. For 4 of the plots, I separated out seeds. And over the course of the season, I tracked bumblebees to get an estimated pollinator behavior. And so what I think is really cool is we have line-based estimates of these traits that have previously been measured. We have line-based estimates of fitness from seed weight from the field. And we have line-based estimates of pollinator behavior. And we can try to kind of link these We can try to link all 3 of these things together to try to understand how color may be modulating selection on these traits, right? So this leads me to my 2 main hypotheses and the 2 main arcs of this talk. The first one I'll be talking about is a pretty simple question. Does color mediate floral selection? Is the presence or absence of one of these colors actually selecting for stuff? So before I kind of jump into that, I'm going to just let you guys know that for a quick sanity check, that we don't see any weird correlations in the lines that we happen to use in the field. Color, as we see on these bar graphs on the left, color, which is on the x-axis, is not significantly correlated with either flower size. In this case, we use overall corolla length as our variable for that, or sugar concentration. And we do see a light correlation between flower size and sugar concentration, but again, this is in accordance with what people have found in the lab previously. So what's going on? Well, this is sort of the meat and potatoes, I think, of like this first part of the slide. It seems like selection on sugar concentration is mediated by flower size. So what I'm showing you here is on the x-axis we have sugar concentration, on the y-axis we have seed set. The pink dots are the pink limb lines, the white dots are the white limb lines, and these are all real estimates for each trait. And you can see that the pink limb lines, as you increase in sugar, you set more seeds, but it's not true for the white limb lines. Now, what I will say, and this is going to be true for a lot of these results, the ANCOVA here is statistically suggestive, so take all these results with a grain of salt, but I still think it's an interesting result that I wanted to share with you all. Now, also, if you note that the area underneath the lines, and I've done this analysis, is the same between pink and white limb lines. So it's not that pink color itself is under selection, it's some sort of modulation that appears to be going on. We see a very similar pattern in flower size, right? So on the x-axis, we have corolla length. The larger your flowers, the more seeds you end up setting, only if you're pink. And again, we have a statistically suggestive p-value via ANCOVA. Okay, so is selection on flower size and sugar concentration mediated by flower color? It seems like the answer is yes, right? For these white limb lines, independent of how much sugar they have or how big their flowers are, they set sort of an intermediate amount of seeds. But in pink limb lines, they set far more seeds when they're more sweet, when they have bigger flowers. So I think this is on its own fairly interesting, but what about what's causing this, right? Can we get to mechanisms with this? And for that, I want to dig into my pollinator data. And I'm really just trying to ask the question here, does pollinator behavior select for sweeter, larger flowers only when flower lines are pink? Before I jump into that, I'm going to just review the different types of pollinator behavior that I'm going to be focusing on for this talk. The first is pretty straightforward. It's visitation. So that just, does a bumblebee like choosing a given line, right? This bumblebee happens to like this pink limb line, happens to like this white limb line. We can make this estimate for each of the lines in the field. The second one is geitonogamy. It's just a fancy term botanists like to use. It's really just sort of a metric of effort. It is the number of flowers that a bumblebee visits when it chooses to visit a plant, right? So the higher geitonogamy means you're visiting more flowers when you're visiting a plant. Okay, so how do these translate to fitness? Well, maybe it's not so surprising. It was kind of a relief to see this, that if you're visited more often, in terms of pollinator visitation, you set more seed, right? More visits you get, you set more seed, which is good, and that's a very strong predictor. It doesn't vary by color. Interestingly, though, for geitonogamy, we see the same sort of interactive pattern. So for pink limb lines, more and more geitonogamous visits mean you set more seeds. For white limb lines, there's no significant trend. So, and this is significant. This difference is significant via ANCOVA. So it seems like there's some sort of modulation of this geitonogamy by color. Okay, so this is sort of the big picture stuff, but we want to kind of understand how this relates to the traits that we're interested in, right? So first, let's think about visitation, right? What are the things that are influencing pollinator visitation, right? So the first thing that we noticed is that pink limb lines are visited more frequently than white limb lines, and lines that have more sugar are visited more frequently than lines that have less sugar. So note here that this isn't a joint effect. There's no, we don't see the same pattern as before, that it's this sugar effect is only happening in pink limb lines. It's just that pollinators like visiting pink lines and like visiting sweet lines. Also note that I'm not showing you a graph here of flower size. That's because we do not see this pattern in flower size, right? The larger you are, it does not impact the number of visits that a line gets. Okay, what about geitonogamy? So geitonogamy, we see a very different pattern occurring, right? There's no direct effects of color or sugar concentration or even flower size. But what we do see, and again, this p-value is hovering around 0.1, right? So, take it with a grain of salt. But we see sort of this cross effect where for pink limb lines, the bigger your flowers are, the more geitonogamous visits you have. But for white limb lines, we see the opposite pattern. So to me, this is kind of interesting because we can sort of think about how pollinator behavior impacts selection and put all the pieces together, right? So For sugar concentration, we have bumblebees preferring pink limb lines than preferring sweeter lines. And the joint effect of these independent aspects of visitation seem to be impacting the fitness, right? That because in pink limb lines, because they prefer pink limb lines and they prefer sweeter lines, we see this trend in pink limb lines, and we're not seeing it in white limb lines. On the other hand, for gytonogamy, we see a very different pattern, right? We see that for pink limb lines, bigger flowers beget more gytonogamy, but we have the opposite pattern in white limb lines. And we also see this interactive effect of color in gytonogamy. So in pink limb lines and only pink limb lines, gytonogamy predicts seed set. And the joint effect of both of these two things together leads to the same patterns of fitness. So even though we have these same patterns of color modulating selection on fitness, it's doing it through two pretty distinct ways that I think is interesting. And I think what's particularly interesting is this sort of mirrors the fact that we have two distinct modules, right, that are only weakly genetically correlated. So just some quick takeaways. Hopefully you gleaned from this talk that the two important flower traits is likely shaped by the presence of a third correlated trait, which itself is not under selection. That pollinator behavior suggests two distinct mechanisms that are leading to patterns of color modulating selection, and that these two behavioral patterns mirror the trait correlations that we saw earlier. Sort of thinking about this more big picture, this may help explain how Selvin syndrome evolved in Ipomoea lacanosa, perhaps a loss of color, relaxed selection on other traits. And lastly, it provides at least some evidence, I think, that a binary trait can lead to correlated selection. And I think that's interesting in and of itself. Here's a bunch of people who I would not have been able to do this research without, and my funding sources, and I'm happy to take any questions. Yes.
So I thought it was interesting that the bees were visiting the lines with more sugar more frequently, but that gytonogamy didn't seem to have an effect with sugar.
Yeah.
How are the bees telling that these lines have more or less sugar? Is there like an honest signal for sugar concentration?
That is a great question. My thought process is, bumblebees tend to trap line, and I'm wondering if they go and sort of sense out which lines are good early on in the season, and then once they've found a good line, they just like visiting that line. The fact that it doesn't correlate with gytonogamy is a little strange to me, because my expectation was that there would be a relationship with sugar concentration and gytonogamy, but we don't seem to really see anything there, so, yeah.
Which is super cool. Thank you very much.
Yeah, no worries. Yeah, Jen. Okay, yeah.
In a similar vein, I guess, I know that in some cases there can be like ultraviolet signaling on flowers that will help lead bees to pollen sources. I don't know if that was anything you looked into, whether the two different colors of flowers have any differences with the ultraviolet patterning, or
That is something that I've meant to look at for a while, and I have not gotten around to, but it is a reasonable thing to ask when we have these colors. There's a whole bunch of other stuff that a can of worms with this entire system I don't really fully want to go into, but there is reason to think that might be interesting to try to at least look for ultraviolet signaling. Yeah.
I may have had a quick look at both ultraviolet and scent, don't seem together in these guys.
Yep.
Can you tell us about the alchemist solving? I remember all these lines did that, but do they all do it in the field? Did you know?
I'm sorry, I didn't hear the first part of that.
Alchemist solving as opposed to gytonogamy solving.
Yes, so these plants do some amount of autogamous selfing. I did not check it in the field, but in the system, selfing rates, like non-gytonogamous selfing rates, are often determined by distance between the anther and the stigma. And in these lines, there is reduction in that distance from the mixed mater. So there's almost certainly selfing that is also occurring in the field. And that's something to keep in mind with, I think in a lot of aspects, but particularly thinking about how this relates to fitness and seed set times. Because if there's a bunch of selfing, that could, if there's lines that are more selfing than others, that could change some of the aspects of fitness, especially those are correlated with other traits.
Hi everyone, thanks for coming. I'm Ariane Chotard, and today I will present you a part of my postdoc researches, which is about the evolution of swallowtail butterfly wing shape, with today a little focus on the hypothesis of a trade-off between aerodynamism and aposematism. So swallowtail is a common name for Papilionidae, which is one of the 7 daylight butterflies families, which count approximately 640 species. The final systematic is ongoing, but close to the end. And this family displays a large ecological variability. For example, you can find all the species in all continents, but also this family display a high morphological diversity. Ooh, my notes are perfect. Nice. Diversity in terms of color pattern, of course, as you can see, but also in terms of wing shape. But the evolutionary determinant of this diversity are still poorly known. But start at the beginning. Start at the beginning. When we think about the evolutionary determinant of wing shape, the first thing we have in mind is flight. Sorry, it's a mess up. Flight. In butterflies, flight is involved in various crucial behaviors, such as feeding, finding a mate, finding a nose plants. And so it's supposed to drive the evolution of wing shape. But the strength of this pressure should vary among the importance of wing parts. And maybe it will work. Yeah. As you can see, butterfly's flight is anteromotoric, which means the butterflies is in a large part propelled by the forewing instead of the hind wings. Dramatic. And so this difference in terms of flight implication should drive in a different way wing shape evolution. Yeah, sorry. So it will be my first question. Do you observe a differential evolution between forewings and hind wings shape? So to answer this question, I worked on the entomologic collections in a large part from the National Museum of Natural History of Paris. And I've taken a lot of photographs for a total of approximately, again, 1,880 individuals, but soon we will reach the 2,000s. And to compare the shapes, of course, I use geometric morphometric tools. To be very quick, geometric morphometrics aims to compare shapes between each other, but we can only compare what is comparable. And so, which means we used homologous point present in all of our shape. It's called landmarks. And yeah, and these points finally describe the wing architecture and wing outlines. So, results. The first thing, I quantified the difference of morphological variability across forewings and hind wings. So to do that, I placed all the shape in the same theoretical space of shape called morphospace. And every spot you can see is a shape. So in orange, it's a hind wing shape, and in blue, it's a forewing shape. And so if we calculate the centroid side of each cloud, it's an approximation of the morphological variability. And the variation of shape is almost 2.70 times higher in hind wings than in forewings. Second, I split the morphospace in two between hindwings and forewings, and I calculate the phylogenetic signal. And this signal is significantly higher in forewings than in hindwings, which means that if 2 points are similar, if two forewings are similar, it's more explained by the phylogeny, the historic of the species, than for hindwings. So third, I calculate the evolution rate for shape, hindwings, and forewings. And again, in the same vein, the evolutionary rate of hindwings shape is 3.8 higher than for forewing shape. And we have the same results for the shift. The number of evolutionary shifts is largely higher in hindwings than in forewings. And fourth, the last, the disparity through time. The disparity through time, called DTT, detects a discernible departure from the null model of body and motion for forewing shape, especially at the beginning of the the evolutionary history, which means that we are less disparate. It's AC. Thank you. I'm not made for the AC. Sorry. So, the disparity of forewing shape is smaller than expected on the neutral model, which suggests a selection around an optimum, but at the opposite side, the disparity of hindwing shapes seems not different than neutral evolution model, so that suggests a neutral or heterogeneous selection. So, to our question, do you observe a differential evolution between forewings and hindwings? Yes, and the characteristic of the evolutionary history of forewings suggests a convergent evolution of shape, potentially driven by stabilizing selection. And in the other side, the characteristic of hindwings shape evolution suggests an heterogeneous selection. This difference of evolution could suggest different selection regimes, which is concordant with previous researches. But if the evolution of hindwings shape is less driven by aerodynamic contrast, what are the other evolutionary determinants? And today I would like to focus on the specific example of predation and how this pressure could affect the evolution of hindwing shape, with the example of the aposematism signal. As we can see, in Papilionidae, there is a lot of species displaying large many traits, many different traits. To be quick, a certain lineage of Papilionidae feed on host plants, known for their toxicity, and the toxins are sequestered by caterpillar and retained in the adult stage. And many of the species have aposematic coloration patterns, making them particularly recognizable to predators. But this color pattern is also associated with some wing shapes, but we don't really know if with wing shape is a part of the aposematism signal. So that will be my my second question, sorry. Do we observe a specific wing shape associated with toxicity in Papilionidae? So to answer to this question, I choose to split to discretize my data, because data of shape are multivariate and continuous, and sometimes it's hard to link a shape with an ecological hypothesis. So to that, I use clustering algorithms to split my hindwings shape data in 2, 3, or 4 clusters. Today I will present this result with 4 clusters. Each cluster is associated with a theoretical mean shape, but these mean shapes are very close to real wing shape you can find in the family. And based on this data, I reconstruct the ancestral state of each theoretical mean shape to see if this evolution is correlated to the acquisition of toxicity. And when we look at the repartition of shape versus palatable or toxic species, we can see that it's not random, but how to do the difference between an association caused to the phylogeny or caused to the selection. And for that, I ran model of evolution, taking into account the disparity of diversification, and the best models, the model which best explanation of the evolution is the correlated model, which means that Heinrich shapes are more associated with toxicity than expected under a neutral model. And so we found an association between Heinrich shape and toxicity. It's not a good one. Okay. Perfect. Sorry. So the first hypothesis which could match the results is Heinrich shape is actually a part of the aposematic signal, but we can explain this result in other ways. For example, in Papiones there is another predator strategy, and it's a possibility that some Heinrich shape are part of another anti-predator strategy, which is only found in palatable species. But I will be very happy to tell the rest of the story about the possible trade-off between aerodynamism and toxicity in Papionidae. But to answer this question, we need the information of aerodynamic performances. And to do that, we are currently working on a computational fluid dynamics model to have this information and to compare the aerodynamic performances and and the acquisition of toxicity. I'm really sorry for my voice. I lost maybe 3 years. Of course, thank you to my co-authors, and thank you for your listening.
So I would like to begin by arguing that the remarkable diversity of plant diversity that we observe around the world is driven to a large degree by variation in life history strategy or life history syndromes. So these comprise, the timing, the environmental sensitivity, the genetic control of life's developmental transitions, such as transition to flowering, how many times you flower and reproduce, whether or not that's determined by the environment, and of course a whole host of other related traits. And so we often describe life history as a syndrome, and I've argued previously that really we should think about the difference between annuals and perennials in plants is really a continuum, right? So there's a whole bunch of covariant and independently varying traits that make up differences in life history, driving life history strategy. And while a lot of past work in the evolution of plant reproductive life history strategies is focused on reproductive traits like, how are meristems controlled through genetics and environment, I've really tried to focus the work that we're doing in life history in my group around thinking about resource assimilation and allocation. I think this is really critical when we're thinking about trade-offs and trying to make arguments about trade-offs to understand what the currency of that trade-off is. What is it that's actually being traded off and that is not infinitely available? And so we've really focused on carbon as sort of a central currency for understanding how these trade-offs occur and how they evolve. And that's nice because it's both the central unit of biomass and energy in plants, but it's also a relatively straightforward resource for us to track as we move through plants. So dating back to the 70s and 80s, plant ecophysiologists have thought in these terms and thought about how plants make decisions about acquiring and allocating resources, a range of different resources. And again, we'll focus today on carbon. So really, the question more broadly for this part of my group is asking whether or not carbon assimilation and allocation really drive life history evolution in plants. And the model that we're using to study this is grasses. So grasses have a wide range of different life history strategies. In addition to the sort of classic C3 versus C4 photosynthesis, we see tremendous evolutionary number of changes between usually perennial ancestors and derived annual lineages. And you can see across the phylogeny, all the green dots are a bunch of comparisons that we've been able to make between annual and perennial congeners. The specific work in my lab focuses on a model grass genus called Brachypodium. These are C3 plants and the Puid subfamily, for those of you who follow grasses. And depending if you're a lumper or splitter, there's between 15 and 20 species in Brachypodium, encompassing a whole range of really interesting evolutionary transitions between ploidy and dependence on rhizomes for growth and lots of other traits. We're particularly interested in the fact that we have two seemingly independent evolutions of annuality in the genus. So in the blue box there's two diploids, Brachypodium stacei and Brachypodium distachyon, that are annuals. But it seems pretty clear that those are both derived from a perennial ancestor, and most of the extant species are themselves described as perennials. The perennials are very broadly distributed through Eurasia, showing sort of a classic, many different latitudes of origin. And as a result, these are very often iteropairs. And we talk about perenniality in Brachypodium, we're talking about a plant that reproduces repeatedly over many seasons, often with periods of quiescence during either very cold or very hot times of year. The annuals, by contrast, are restricted to a relatively narrow distribution right around the Mediterranean, so they're experiencing sort of a classic Mediterranean climate with relatively cool, moderate winters and very hot, dry, probably lethal summers. And accordingly, the annuals in Brachypodium are best thought of as being semipairs, right? So these are plants that are behaving much like Arabidopsis does. They're probably germinating in the fall, overwintering, and then reproducing and setting seed before the terminal summer heat and drought. So this is a nice model system for us to address some really classic questions in the origin and evolution of life history strategies. So classically, G.C. Williams and others, have posited, trying to understand what the costs of these rapid versus slow growth might be. And I want to focus on this one classic idea that Williams argued if the chance of surviving to a second bout of reproduction is sufficiently low, then selection for increased early vigor may have few trade-offs. And Garnier, who is a grass ecophysiologist developmentalist working mostly in the 90s, did a lot of nice work sort of extending this, arguing that selection for increased growth rate in short season environments favors alleles that increase early vigor at the expense of longevity. And so a lot of these early ideas were building from arguments in animal systems where perhaps there was this sort of energetic cost or a damaging cost of having very high growth rate, but again, placing this in the realm of metabolism and allocation and thinking about how organisms are using, in this case, their carbon resources. So really, this is talking about the interaction between acquiring carbon and how that carbon is used for growth or respiration maintenance. So the first hypothesis to test today is that annual Brachypodium have a higher rate of carbon assimilation and a higher relative growth rate than they do the perennial congeners. And I don't have a figure for carbon assimilation. To take my word that they do have a much higher rate of assimilation. Looking at the two annuals in our system and the one perennial, we can focus on the y-axis is relative growth rate, so this is the amount of biomass you're putting on per day, per your unit mass, so a nice way to sort of size standardize things. We see that both annual species have a higher relative growth rate than does the perennial in this study. Okay, so the next question to address really gets at this issue about how are you investing your resources early in life? And so we hypothesized that given a stress, that our annual Brachypodium might sacrifice future survival to make sure that they can actually get some reproduction out in this with their one shot in this 1 year before the terminal drought. So now trying to link carbon acquisition, growth and maintenance to reproduction. So to address this, we did a gradual soil drying experiment. I'm going to use this figure to sort of outline when these measurements were made and give you some context. I just want to highlight that this is the work of my PhD student, Jia Yun, who is phenomenal, the data sets she's been able to put together. She's also looking for postdocs, so reach out. But she's a great physiologist and also now become a really good computational biologist. So as we go along, I'll try to highlight when these measurements were made. And the real key point is that the plants were grown vegetatively for 21 days in the case of the annual, a little longer for the perennial because they flower later. And right at the beginning of reproduction, we started tracking individual seeds, but also started making a whole series of physiological measurements along the soil drying treatment. So the first thing I want to highlight, and I apologize, this is really difficult if you haven't looked at carbon assimilation data before, we're just looking from left to right is basically the first 8 days of the soil dry down. The blue bars are control conditions, looking at the rate of carbon assimilation on leaves. So this is, what's the net carbon coming into the plant. And the orange dots are the dry down, looking at the same trait. So again, if you haven't looked at these before, in the gray bars, you're looking at nighttime, and so then we have a net production of CO2. The plants are respiring, not fixing any carbon, so we expect to see those negative values in the brown parts. What I hope you can see is that as you move towards the right, as the plants get drier and drier, the carbon assimilation in both plants is basically moving to 0. And this has a significant impact on their carbon status. So this is a measurement of leaf starch, so a nice carbon storage molecule. Plants tend to use the carbon that they need and store what they don't. And here's a case where it seems like they don't have any extra, right? So the leaf carbon is getting reduced under drought stress. Okay, so the carbon status of both plants has changed, both the annual and the perennial. What does that mean for life history? So Gia tagged a bunch of seeds at the first day of the stress and came back 10 days later after the seeds should have completed their development. Okay, so these are now presumably mature, healthy, dry seeds. And the contrast I want to make is on the left, you're looking at the annual and the perennial top and bottom, looking at day 0 when this process started, so you can see that both species are filling their grains as expected. And if you contrast the blue and the orange bars over there in the annual, you see that there's very little and no significant difference in the individual seed masses of drought stress versus control annual brachy. But in the perennial species, we see under control, they continue to fill their seeds nice and properly, but there's a statistically significant decrease in the seed mass under the drought stress. Again, tracking the carbon, this is looking at seed respiration. So as plants are filling their seeds are generating CO2 because metabolism is happening. And so here a more negative value indicates higher respiration, more physiology going, more metabolism going on. And again, we can see that the annual shows no significant difference between the treatment and the control, whereas we see considerably more respiration happening in the control perennial seed than the drought perennial seed. So they're growing more slowly, and we can see that both phenotypically and looking at gas exchange. Okay, so it seems that perennial Brachypodium is sacrificing its current reproduction under this drought stress. So what is it doing with the carbon instead? Well, this is a drought stress, and one thing that many herbaceous plants will do under drought stress is invest in the resource that can help them get more of that resource. So we often see increase in root-shoot ratio. This allows plants to explore more water, but also decrease their water consumption above ground by reducing that ratio. Again, looking at the annual first on top, we see that there's no significant change in root-shoot ratio as a result of this drought stress, or a very minimal, non-significant one. By contrast, the perennial is increasing its root-shoot ratio. It's prioritizing establishing that base of resource acquisition. And so it seems that the annual here, the rapid growing annual, is sacrificing the prospect of future resource acquisition when faced with the drought stress. So, to summarize sort of the central part of this talk, annual Brachypodium is prioritizing the semelparous reproductive output, while the perennial seems to be prioritizing sort of the long term, right? So maybe like a bet hedging kind of a strategy. So just sort of graphically summarizing that. So this led me to think a little more deeply, sort of trying to link physiology now to development. And we were wondering, well, if just annual Brachypodium is less developmentally plastic, right? If its resource changes, if resource availability changes, perhaps it just can't adjust as readily at least in a vegetative stage. So to test this hypothesis, we did a different experiment, and this was the work by my former postdoc, Marge Lundgren, who's now faculty in the UK. And the experiment here was to take 2 annuals and 2 perennials and to expose them to a range of atmospheric carbon dioxide. So this is effectively a way of limiting the carbon source by scrubbing CO2 out of the atmosphere. So this is, sort of below pre-industrial CO2, a 400 parts per million control, and then a vast excess. So not trying to replicate climate change, but just trying to juice the system with a lot of carbon and asking if the plants can respond developmentally. So we're going to make it 3 different species in these 3 different treatments. So first, just to convince you that juicing the system does in fact do that, right? So as you increase CO2 and looking at the 3 species at 180, 400, 1500 ppm, you can see that all 3 species are increasing the rates of carbon assimilation as we're giving them more CO2 in the atmosphere. So that's good. So we're adding carbon to the system. So what do they do with it? Here we're looking at relative growth rate on the top, so the same trait I showed you earlier, this whole plant measure of growth rate. And the contrast I want you to focus on is between 400 and 1500, so control versus lots of extra CO2. What I hope you can see is that neither annual shows any response to this extra CO2. Their relative growth rate is unchanged, whereas the perennial is dramatically increasing its growth rate in response to higher CO2. And this is driven almost entirely by production of what we call tillers in grasses. These are branches, right? So these are novel innovations that allow the plant to basically acquire more and more resources, the more leaves and more roots, and ultimately, we think more reproduction. Okay, so just to wrap this all up, I've shown you today that annual Brachypodium establishes quickly. It's a rapid growing plant. It's probably trying to grow quickly in this short environment, and it seems to prioritize reproductive output in a stressful environment. By contrast, the perennial Brachypodium, while still filling seed, is doing so a lot less. Perhaps it's investing in sort of the ability to see a second season, so there's sort of a bet hedging strategy. We've gone on to show that annual Brachypodium shows relatively limited plasticity in plant architecture or whole plant growth rate. And I'll just say that a lot of this is really sort of the background work to try to get at the regulatory basis of this, both within Brachypodium and then ultimately across the lineages of other grasses where we see transitions between perennial and annuality. So I'll just acknowledge my current and former research lab members, funding sources, and a wide body of group of researchers helping with genomics and comparative biology. Thanks. Looks like I do have a minute for a question. Yeah, Linda.
This may be an off-the-wall question that you don't think about, but how do these two perennials versus annuals compete, given the life history differences?
With each,
Speaks of who?
with each other or just in their community?
Other.
I am not aware that, so, I showed the distribution of the perennials as being this very broad thing. I don't believe that they co-occur in the habitat in the Mediterranean. I can't say that exclusively, but they do seem to have some niche partitioning among them. Even the two annuals have a bit of a partitioning along an aridity gradient. So I think the Sylvaticum are further inland usually. Yes?
What kind of soil did you use for growing these grasses, and were they fertilized?
Yes, they're absolutely fertilized. So when we're doing our whole plant stuff where we want to be able to look at root anatomy and morphology and so forth, we actually grow them in a fritted clay media, so it's very easy to wash off. And we've done side-by-side comparisons. They're happy to grow in that. But we're providing all their nutrients, and they're nutrient replete in all of these. Yeah. All right. Thank you.
So we have a lot to cover, but I'm looking at phylogenetic comparative methods for analyzing function-value traits. And I want to start off by asking, how should we be considering our traits? How are we thinking about them? And we know that all ecophysiological traits, which can determine how a species is going to interact with the abiotic and biotic environment around them, can be described as functional traits. And in the plant sphere, some common functional traits are leaf mass area, vein density, anthocyanin content. But if the trait that you're interested in is not independent, or if it perhaps fluctuates as a dynamic response to the environment, it might be more appropriate to think of it as a function-value trait, which is modeling this trait, this response, over a continuous interval. And by representing our response as this continuous function over some infinite range, we can interpret it as a function-valued trait, which has a few advantages in analyses. So by normalizing our response across species with a mathematical model like nonlinear least squares, we can account for linear variation between species, and we can quantify the plasticity in that trait as the magnitude of change in our predictor variables. We can then extend this function-valued trait approach into phylogenetic and comparative analyses in order to make phylogenetically informed predictions of the function-valued trait, test for phylogenetic signal in the response as a whole, and potentially even make imputations and predictions about how this response changes in species that you haven't been able to measure. So for those unfamiliar with photosynthesis and this dynamic response, it's often measured by light response curves, which measure the change in carbon dioxide assimilation over a range of incident light intensities. And the linear behavior of this light response curve can describe photosynthetic processes which are limited by light availability, limited when light is saturating, but is limited by carbon availability, and it can even detect a decline in assimilation when the leaf is overexposed to light and there is damage to the photosystems inhibiting photosynthesis. Now, once we've established this function-valued trait, we can extract functional traits from this response for the basal metabolic rate, or the dark respiration, light compensation point, light saturation point, the quantum yield, or the rate of photon capture, and the maximum net assimilation, which we can then use to better compare between species. Now, we chose to focus on the genus Helianthus, which has an adaptive radiation across the North American continent about 3.5 million years ago. There's a lot of extensive molecular data in this system. So there's a well-resolved phylogeny and a fully assembled reference genome for the species Helianthus annuus. Individuals in this genus have different life history strategies, so annuals and perennials. They have very diverse ecophysiological traits, and they occupy a broad variety of habitats across the continent. So that includes deserts, marshes, forests, prairies. And as a bonus, species in this genus, species like Helianthus annuus, are important agricultural crops. The third largest oilseed crop in the world, which has led us to help establish this system as a model system for phylogenetic comparative analysis in plants. So in our study, what we did is we grew 28 species of non-hybrid diploid wild sunflower, and we took light response curves on each of them at 9 levels. We then modeled those light response curves with nonlinear least squares into function valued traits, and then extracted traits and used those curves themselves in phylogenetic comparative methods. The first step here being the function value trait construction, and so we tested 9 different nonlinear models in order to reconstruct our trait, our response. And you can see here some of the variation between species, I'm showing the variation between species, but more what I want you to focus on is differences between how each equation modeled the curve. Each equation has slightly different assumptions about trait relationships, even though they all consider the physiology and biochemistry between them. And you can see some differences, especially in the curvature of the line where it plateaus, the increase in the line where we see quantum yield, and in equation 11, you can even see one tiny incidence of photoinhibition detected. So we evaluated all of these models with mean squared error and saw that equation 6, the empirical non-rectangular hyperbola model, was best fit for our data. And I want to stress how important it is to test these different models for your light response curve data, because we get different answers with each one. It seems to be pretty underreported in the literature, but the people seem to just choose one of their favorite models, which is really not the best way to go about it. So you may see an R package to address this soon. So once we had these function valued traits, we started to ask ourselves, how are we actually going to calculate our trait values from this? And we took sort of a multiverse approach where we tested everything and then are going to report everything to you. So if you take your light response curve data and then make a function value trait with that, you can either take a trait-based reconstruction approach or a curve-based reconstruction approach. And for the trait-based approach, what you do is you can take physiological traits like dark respiration, the basal metabolic rate, quantum yield, light compensation, directly from the curve and put that into evolutionary models so that you can detect phylogenetic signal in each trait. Alternatively, you can take those traits and find correlations amongst them because they are not independent of each other, and then use that to better inform your evolutionary models and find consensus phylogenetic signal in response. Or alternatively, we tested this new approach for the curve-based reconstruction, where we, after fitting the function value trait, we found descriptor parameters of the curve to describe the linear behavior of the function valued trait, where we described the assimilation as a percentage of light exposure. So we can model the behavior at the beginning and the end of the light response curve really well, and then hopefully, then we find correlations between that, inform the evolutionary model, and hopefully make a better prediction of the light response in totality. Just as a check, a point of comparison to make sure that we weren't looking at, we weren't trying to interpret measured variation in our data set, we also took a non-function valued trait approach where we just took the measured carbon assimilation values at each measured light intensity and treated that as a trait, and then put those either in a univariate model or found correlations amongst them and put it in a multivariate model to test for evolutionary signal in each measured value. And that's kind of what you're looking at here, where we have the phylogenetic signal in carbon assimilation as a function of light intensity, and each point represents our non-function valued trait approach, where it's just measured data, or phylogenetic signal in the measured data. And then the smooth line is the phylogenetic signal across each light intensity as a function valued trait. And overall, there is a lot of agreement between these models, except in the very early part of this figure where there is discordance between phylogenetic signal between 0 and about 250 PAR. And unfortunately, at these light intensities, that's where we see some of the most evolutionarily labile traits, being basal metabolic rate and the light compensation point. Now, of course, this could speak to the inter- and intra-specific variation amongst these species. But I think it also speaks to the back and forth evolutionary dance between these species for these traits as they adapt to different environments. It's also worth noting on this slide that from our model, we see very strong agreement in phylogenetic signal in photosynthetic response at medium and high light intensities, which tells us that these responses at medium and high light intensities are well conserved across the phylogeny, and that there are clade-specific responses to medium and high light intensities in photosynthesis. So there are a lot of benefits of using each of these approaches, but I think ultimately they work. I think ultimately the right approach really depends on the question that you're trying to ask, and to some extent the model that you select to construct your function valued trait. I guess I'm trying to ask, if your question is to just survey the variation in univariate traits, a univariate model is fine. But if you want to look at a more comprehensive view and study how the response changes as a whole between species, you should look at the multivariate models, or preferably use both of them in tandem. So that's what we did. The first figure at the top is showing just the function valued trait constructions of each species mean for annual in red, perennial in green, and erect perennial in green, and basal rosette perennials in green, erect perennials in blue, annuals in red. And you can see it's a little messy. When you take that data and reconstruct it with our curve-based approach, you can see that there's a shrinkage in the noise that we observe. And then once you further that by incorporating the established phylogeny in our evolutionary models, we see a very nice clustering of these 3 different growth forms within the phylogeny. And of course, our analyses show that there was a strong effect of phylogeny on the overall photosynthetic response. And so to tease apart what functional traits were actually driving that significant variation, we used the univariate trait approach to look at phylogenetic signal across each trait. At the bottom, I have the basal metabolic rate from left to right, basal metabolic rate, light compensation, quantum yield, and maximum assimilation. We only saw significant phylogenetic signal in the maximum assimilation value, but we did see some pretty moderate signal in the quantum yield, and again, just too much variation in the light compensation and dark respiration to attribute that variation to phylogeny. And ultimately, we did these exhaustive analyses in order to bring some conclusions about which trait approach to use. We saw that the multivariate models, the trait-based approach and the curve-based approach, were essentially equivalent. The prediction values and the imputation values for these traits were identical, but there was slightly stronger support for imputations with the trait-based approach. Interestingly, you found better imputations with the trait-based approach using the empirical models for function valued trait, but this support changed if you used the mechanistic models. The mechanistic models tended to work better with the curve-based approach, but they both gave really great data, and using this curve-based approach gave us better descriptors of how the curve is actually functioning across light intensities. We found some really interesting correlations between environmental traits and physiological traits from the values we extracted in this data set. And I just want to touch on some of them briefly. Ultimately, I think for many of the traits, we found better correlations with our evolutionarily predicted traits. You can see in the first two graphs, maximum assimilation with soil pH. The predicted value had just slightly better, it just narrowed the noise, reduced the variation in the distribution of those correlations. But we also found correlations with a lot of our variables with environmental things like precipitation, seasonality, pH, of course, aridity, and also correlations with some physiological traits. One of the most interesting to me was correlations between quantum yield and production of defense compounds, like you see in that last figure. So ultimately, you should use both of them in tandem to get a holistic view, understanding of the change in these traits and their response. And this approach can be used for almost any system, adapted to every system, and you'll see code for that shortly. With that, thank you to my lab, to my advisor, Dr. Eric Gouldsby, and to all of you for joining me. Do we have time for questions? Is anyone curious? If not, you can meet me in the coffee. Yeah.
What were the ambient light conditions in the greenhouse? Were they pretty bright?
So we did this study in Central Florida. It was pretty bright. That picture was, I came to water them at night one day, and I just had a nice shot. But I would say at least 1,000 PAR. Yeah, but like I said, these species come from all over the United States and North America, so most of them are adapted to very, very high light conditions. I think some of them would have been happier in even lighter conditions. Thank you.